The overall goal of these procedures is to quickly isolate highly purified neutrophils from human blood samples by density gradient centrifugation to investigate their role in lymphoma cell sensitivity to cancer therapy using 2D and 3D cell culture models.
These methods can help answer key questions about the interactions between the innate immune system and cancer, such as what role does neutrophil-mediated tumor cell responses play in cancer therapy. The main advantage of density gradient separation is the ability to isolate pure immune cell populations in a short period of time without exposing the cells to function-altering stimuli.
To isolate the primary leukemic cells and neutrophils, begin by adding 15 milliliters of EDTA-treated blood from chronic lymphocytic leukemia patient samples into individual sterile 50 milliliter tubes. Dilute the blood with 15 milliliters of rPMI. Then, carefully and slowly, add 15 milliliters of room temperature density gradient solution under the blood, taking care not to mix the layers. After separating the cells by centrifugation, 4 distinct phases will be visible: the platelets and plasma at the top,